Started. Yeah, good morning to all of you. So let us see the remaining part of what the transcription. Now we are discussing about the process of transcription. Yesterday I told you the different steps in the process of transcription. Yeah, there are 3 steps in the process of transcription, like initiation, elongation, and termination. Initiation, you may be remembering, it's just for the starting of the new strands of RNA, and the termination is what the ending process, and elongation is in between the promoter and the terminator only. So this is what the things yesterday we have seen here. Then initiation with the help of the sigma factor present in the promoter region of a transcriptional unit, and elongation is because of what the RNA polymerase, DNA dependent RNA polymerase, it is moving along the length, and 3' 5' strand of the DNA only acting as what the template here, because the movement of what RNA polymerase always from the 5' to 3' direction, and after reaching to the terminator region, and this because of this raw factor, it's separated, RNA polymerase separated, with that the process of the RNA preparation will come to end, and this will detach, and it will come out from the DNA, will go for coiling and getting that 3-dimensional coiled structure back. And this is what the initiation, and I think you already discussed about it, and is what the elongation, and next one, that is what the termination. It is the process to stop the elongation of the RNA strand only, and once the RNA polymerase enzyme reaches the terminator region, as I told you, that piece of RNA falls because there is a factor present there. We call it as what the raw factor, and this raw factor only help the RNA polymerase to detach. So because of what the absence of RNA polymerase, it is not possible to elongate further. So with that, the process of the transcription will come to an end, right? Then this RNA that produced from this DNA through this initiation, elongation, and termination is called as what the rhRNA, or we call it as heterogeneous nuclear RNA. Tell me where this process of what the transcription take place in a cell. Of course, it is in the nucleus only, because DNA or the chromosome present only in the nucleus. So naturally, this process take place only in the nucleus. That RNA synthesized from the DNA is called as what hnRNA, and it is really a raw RNA only, and it is not at all processed, and it is not ready for the next translation process, because this RNA consists of lot of unwanted sequence of nitrogenous bases that is not at all required for the production of protein in the cell cytoplasm. So it should be corrected, and it should be corrected by eliminating all the unwanted sequence. And this process of making RNA, mRNA, a matured RNA is from the hnRNA is called as what the processing only. So there are 3 steps in the processing of this hnRNA, and that only we are going to discuss here. That the capping is there, then splicing is there, and last tailing is also there. So after capping, then splicing and tailing, only this hnRNA will be converted into mRNA, and all these 3 steps of processing of RNA take place only in the nucleus. And after this formation of that matured RNA, this RNA will be taken from the nucleus to the cytoplasm for what you can say that the last process, we call it as what the translation. So that only we are going to discuss now. RNA transcribed from the DNA is not able to use for translation directly, and it is called as what the hnRNA only. It's purely a raw RNA. It should be matured. because it consists of some unwanted nucleotide sequences. Unwanted means this unwanted only for that particular protein. Maybe it's required for some other proteins. So depend on the protein only, we will tell this is what the unwanted and this is what the wanted sequence of nucleotides. And for producing some protein, this sequence is not required. And for producing some other protein in the cell, sometimes the sequence is required. That is why they are keeping all the sequence in the RNA. So whatever the sequence of our nitrogenous base are not required for producing a particular protein, that should be removed. And such a type of unwanted nucleotide for production of a particular enzyme present in these RNA is called as what? The intron, unwanted sequence, and the remaining wanted sequence or the required sequence of nucleotide for producing that protein is called as what? The exon. Remember these two terms, intron and exon. In a unprocessed RNA, we call it as what? hnRNA. So many sequences are there, and some key sequences are not required for a particular protein. We call it as what? The intron. And some sequences are very much required for producing protein. We call it as what? The exon. If introns are not removed, it is not possible to produce the functional protein because some amino acids will be there in the protein. It totally disturbs the sequence. So naturally, that will not work in the cell as what? The functional protein. So HNA RNA also consists of what? The wanted nucleotide sequence, and it is called as what? Exons. So for translation, the defected non-functional hRNA should be converted into a functional matured mRNA. And this is called as what? The processing. Conversion of the hnRNA produced in the nucleus of the cell should be converted into what? A functional mRNA. And this process is called as what? The processing of RNA. And there are three steps are there in the processing. One is what? The capping, another one is tailing, and another one is what? The splicing. We will see. Before that, let us see some of the differences between that transcription in prokaryotes and transcription in eukaryotes. So remember, in case of prokaryote, there is no nucleus. Well-defined nucleus is not there. Whereas in that nucleus is there. So everything, every process of what? The transcription happening, even processing is also happening inside the nucleus only. But in case of what? Such a type of the well-defined nucleus is not there. It is having only the nucleoid, and this DNA is exposed to the cytoplasm. Let us see how this process of this transcription happening in the prokaryotes. See, RNA does not require any processing in prokaryote. That means everything is necessary. All are exon, no introns. It consists of only exons and no introns in prokaryotes. And prokaryote transcription and translation take place together. Why it take place together? Because in case of eukaryotes, you know that inside the nucleus only, what happened? The process of transcription take place. Whereas in case of what? The prokaryote, such type of nucleus is not there. So transcription and translation can take place together because no boundary for the transcription and translation. So that is why in case of prokaryote, transcription and translation take place together because no nucleus. Then translation can start much before the completion of transcription, that is there in prokaryote. Transcription, process of what? The RNA from the DNA happening in the nucleolar region, and no need to process it. So what happened after making some RNA and it come outside from the DNA, immediately what happened? The translation also start. So whenever it stops the transcription, immediately the translation also will be completed. So this is what the things happening in case of what? The prokaryote. Transcription and translation can couple in bacteria like a prokaryote. But in eukaryote, translation take place only in the cytoplasm, only after the completion of the transcription in the nucleus. So these two are two different process in what eukaryotic cell. Transcription take place in the nucleus. After the transcription, it should be processed. Then only it come to the cytoplasm. Only inside the cytoplasm, the real process of what the translation take place. And in prokaryote, only one RNA polymerase. I told you yesterday, in case of eukaryotes, there are three type of RNA polymerase, that is to synthesize three type of RNA: rRNA, tRNA, and mRNA. RNA polymerase 1, RNA polymerase 2, and RNA polymerase 3. Whereas in case of prokaryote, one RNA polymerase is enough for synthesizing all the three different type of the enzyme, all the three type of the RNA, tRNA, rRNA, and eukaryotes. Then that you have to remember, because many times it's asked in the examination. So come to what the RNA processing. How it's go for processing. Processing means what? Conversion of hnRNA, heterozygous nuclear RNA, into a matured RNA, because only the matured RNA can go for translation. Matured RNA only give the correct information about the total number of amino acids present in the protein, and also giving the correct information about the sequence of amino acids present in the protein. That only determine the quality of the protein. That function of the protein is mainly determined by these two things: total number of amino acids present in the protein, and the sequential arrangement of amino acids present in the protein. Of course, that structure, primary, secondary, tertiary, or quaternary structure of the protein can also determine the function of protein in the cell, as an enzyme or carrier protein, or what you can say, this contractile protein or enzymes, etc. Then come to the process of processing of RNA. So this is to convert hnRNA, unprocessed or raw RNA produced in the nucleus, into what the functional mRNA, a matured mRNA. mRNA is not matured mRNA, it is what the messenger RNA. M stand for messenger. So mRNA is matured one, and it is giving all the information related to what you can say, the protein, the total number of amino acids and the sequential arrangement of protein, I mean amino acid in a protein. And that is why it is called as messenger, because it is giving message regarding what the total number of amino acids and the sequence of amino acids in the protein. There are three steps. As I told you, capping, splicing, and tailing. So it is to convert primary transcript. Primary transcript is what the hnRNA only. It is a raw material into secondary transcript, that is what the mRNA. Primary transcript is what? The hnRNA, because it is produced directly from the DNA. That is why we call it as what? The primary transcript. And this undergo the capping, splicing, and tailing. So this will be converted into what? The secondary transcript. That is called as what? The matured RNA, or this messenger RNA, or mRNA. Then RNA processing takes place only in the nucleus. There are three steps in the process of the RNA processing: capping, splicing, and tailing. And all these processes are happening in the nucleus only after the maturation. What happened? This RNA nucleus to the cytoplasm. So let us see what is capping. Capping is always in the 5' direction of the RNA only, because RNA synthesis, hnRNA is synthesized in the direction 5', 3' direction. Immediately after starting the process of transcription, 5' of the hnRNA comes out from the double-stranded DNA. Here you can see this, what the process. So this is what the process of what the RNA synthesis, and it is always elongate new RNA from 5' direction to 3' direction. This is what the elongation, direction of elongation of the new RNA. And whenever this 5' end comes out from this double-stranded DNA. Naturally, this attached with guanosine methylated guanosine triphosphate molecule in the 5' end, and that only we call it as what the cap. It is called as what the cap. Here we can see that the capping process. So immediately after starting the transcription, 5' end of the hnRNA comes out from this double-stranded DNA, and this end of the hnRNA, you see that the 5' end only comes out first, bind with the methyl guanosine triphosphate molecule. That is methyl guanosine triphosphate molecule. It's connected to the methyl guanosine triphosphate molecule. That is in the 5' end of RNA. And it is called as what the cap, because this is the head region, and this end is what the tail region of this newly constructed RNA. So in this 5' end head region only, this methylated guanosine triphosphate molecule attached, and it is only called as what? Its cap, and process is called as what? The capping. It is called as what? The capping only. And process of capping take place even before the termination of the transcription. Immediately after taking that 5' end of RNA from the DNA, it go for this capping process and add that methyl guanosine triphosphate in the 5' end of this newly constructed RNA, and it is called as what? The capping. Then come to the splicing. As I told you, there are two types of sequence present in immatured hnRNA. Some sequences are required for protein synthesis, and some are not required. And required sequences are called as what? The exon, and unwanted sequence present in the hnRNA is called as what? The introns. So this process is to remove unwanted sequence of intron from the hnRNA molecule, and it is called as what? The splicing process. So it is the process to remove unwanted sequence called introns from the hnRNA to make it matured and meaningful. After removing all the introns from hnRNA, all the wanted sequence, exons only are joined together in order to form a continuous RNA strand, and that become a meaningful one. Meaningful means it is giving the complete information regarding the formation of a new functional protein. How it's go for joining? Always remember that glue, biological glue, and it is what? The ligase enzyme only. Ligase enzyme only helping the DNA fragment after removing the introns. Exons should be joined together. That is with the help of the ligase enzyme only, and all the unwanted sequence will be removed through the pinching process. Here you can see that process. That red colored one is what? The introns only. These are all unwanted sequence only, and with the yellow colored one, it is given that exon. These are all exon, and the red colored ones are the introns only. How this splicing take place? It will go for looping, and it pinch off from this RNA. So this is what the way through which all the introns are being removed from the hnRNA and getting what? A matured RNA here. That yellow colored one, that is what? The matured RNA only. There is no intron, only the wanted sequence or the exon. All the pieces, after removing everything, it will be converted into what? Small pieces that can be joined together with the ligase enzyme, and this process only we call it as what? The splicing process. Then come to the last process, that is what? The tailing only. And this is the process to add adenylate residue at the 3' end, and it is happening only after completing that process of transcription. So entire primary transcript comes out from the DNA molecule, and in the 5' end, the capping will be there, and the next end, that is called as what? The tailing. That is what? The 3' end of the hnRNA only, and there in 3' and add many adenylate residues. around 200 to 300 such residues will be added to the tail end, and that is why we call it as what, poly A tail. We call it as what, the poly A tail. Why it's called as what, the poly A tail? Number of residues will be added to the 3' end, and this residue is what, the adenylate residue only. That is why we call it as what, A, poly A tail. A stand for the adenylate residue only. Around 200 to 300 such type of polyadenylate residue will be 3' end of the primary transcript, and this process only we call it as what, the tailing process. Here that tailing here, polyadenylate tail, polyadenyl residues, added 200 to 300 number in the 3' end. It is given in this green color here, right? And this process is called as what? And this is what, the polyadenylate tail, and this process only we call it as what, the tailing. So there are three processes here. One is capping, and that is in the 5' end. Then another one is what, the splicing, and see the removal of the intron from the exon, and another one is what, the tailing at the 3' end only. Then only this what you can say, hnRNA, a raw RNA produced directly from the DNA, will be converted into a matured messenger RNA, because it is only giving the correct message to the machinery present in the cytoplasm to prepare a particular protein having a definite function. It may work in the cell as an enzyme, or it work in the cell as a hormone, or it may be carrier protein, or what you can say, or contractile protein. There are so many type of proteins present in our body, and all these proteins we are preparing through the same mechanism only. So here, this is what, three process: capping, then splicing, and tailing. And after capping, splicing, and tailing, the RNA is called as what, the mRNA, messenger RNA, because that only giving message for construction of a new meaningful protein. And this mRNA is transported from the nucleus to cytoplasm through a nuclear pore. So many pores are there in the nucleus, annulus. Through this nuclear pores only, this matured RNA produced or processed in the nucleus is taken to the cytoplasm, and this mRNA only used in the cytoplasm for the real process of protein synthesis. That only we call it as what, the translation process, right? So come to the process of translation. Tell me where this process of translation take place. No doubt it is in the cell cytoplasm only, because transcription in what you can say, in the nucleus, because the material required for the transcription, all the enzymes, or what you can say, DNA, all the things are there only in the nucleus. So the transcription process take place in the nucleus, and after the maturation and the processing, it is taken to the cytoplasm, and after coming this mRNA into the cytoplasm, the real process of translation, protein synthesis take place in the cell cytoplasm only. Why the translation in the cytoplasm? Because lot of machineries are required protein in the cell cytoplasm, and it is available only in the cell cytoplasm only, not in the nucleus. So what are the machineries required for synthesizing protein by using this RNA? And it includes what, the rRNA produced in the nucleus and taken to the cytoplasm, and tRNA produced in the nucleus and taken to the cytoplasm, mRNA produced in the nucleus and through the process of transcription and taken into the cytoplasm. And another one is what, the amino acids of 20 different types. Amino acids are abundantly available in the cell cytoplasm, because it is getting from our digestive system whenever we take the protein-rich food material, and this protein will undergo the process of digestion, and ultimately it converted into amino acid, and some amino acid can also be synthesized in our body. And this amino acid ultimately taken to the cell, so it is available in cell cytoplasm. That is why we call this cell cytoplasm as what? The amino acid pool. Lot of amino acids are there, and many amino acids are there of 20 different kinds. There are 20 different type of amino acids present in the cell of 20 different types. With these 20 different type of amino acids only, we are making thousands of varieties of protein. How it's possible to make thousands of varieties of protein with this 20 different amino acid, only with this 20 different amino acid? It is easy. How we are making many number of words with a limited number of letters, alphabets in English? Just to change the location, just to change the number of what this amino acid, and change what the sequence of amino acid. So with that, we will able to make, our cell is able to make thousands of varieties of protein in cytoplasm. So that only we are going to discuss here. These are the machineries. One machinery is what? The ribosomal RNA, or we call it as what? The rRNA. It is for the formation of polypeptide chain. Polypeptide chain means what? It is chain of amino acid connect each other with a peptide bond. What is protein actually? A long chain of amino acids only. One amino acid connected to another amino acid, then like that. 20 different kinds of amino acid we have. According to the nature of the protein, the cell is arranging the amino acid one after another and connect each other with a bond, peptide bond, and this long chain of amino acid connected with the peptide bond is called as what? The polypeptide chain. And this rRNA only bond formation between the amino acid during the process of protein synthesis. And why it is called as ribosomal RNA? Because this RNA is placed in between what? The large subunit and the small subunit of RNA. There are two subunits in a ribosome. One is small subunit and another one is what? The large subunit. And the large subunit is placed over what? The small subunit, and there is a gap in between, and there in between these large and small subunit only this rRNA, ribosomal RNA, is located, and this rRNA only responsible for connecting all these amino acids in a correct sequence. See, methionine, proline, valine, arginine, lysine. These are all what? The amino acids only. All the amino acids connect each other with what? The peptide bond, and it is produced because of what? The rRNA present in the ribosome only. We call it as what? The ribosomal RNA. And it is very specific, and you know that ribosome is what? The site for protein synthesis. Why it's became the site for protein synthesis? Because of what? rRNA present in between the large subunit and small subunit only. These are the this is what? The ribosome, large subunit and small subunit. In large subunit, we can find there are three sites. We will see the significance of the site present in the large subunit later while discussing about the process of translation. And there are three sites: A site, P site, and E site. A site is called as what? The acceptor site. You may be remembering that the tRNA will be accepted by the A site only. Then from the A site, it will be taken to the donor site. And from the donor site, P site, it will be taken to the exit site. And from the exit site only, this will be taken out. See, whenever this ribosome move through this what? The RNA, it is called as what? The mRNA. This is mRNA present in between what? The large and the small subunit. And whenever this what? The ribosome move at the time of translation, so naturally what happened? This tRNA will place the amino acids according to the information given in the mRNA one by one. That is what the reason for the formation of the polypeptide chain. And acceptor site, that is what amino acid, or it is what the binding site, we call it as where A site only receive the tRNA. Then peptidyl site or binding site is what the P site. It is the donor site only because it donates this tRNA to the E site. And why it is called as E site or exit site? Because whenever it moves from the exit site only, the tRNA the cytoplasm. Then this will go after activation, this tRNA will go and pick up another amino acid and place one after another according to the information given in the tRNA, I mean mRNA only. That is why we call it as messenger RNA. It is giving all the message related to what the sequence of amino acid present in the polypeptide chain, and also determine the total number of amino acid present in the polypeptide chain. These are the three sites: A site, then P site, and E site. A site to receive, it's acceptor site only. P site is donor site. E site is what the exit site. From here, it will take this tRNA out, and it is large subunit and small subunit. And through this small subunit only, this what mRNA, messenger RNA bind. That is why we call it as what the mRNA binding site. Then come to the next machinery present in the cell cytoplasm for translation. That is tRNA, transfer RNA. Why it is transfer RNA? Because this RNA only pick up the amino acid from the cytoplasm or from the amino acid pool and taken to the site of protein synthesis. So amino acid pool cytoplasm, lot of amino acids are available. So they have to find out the correct amino acid to be placed in the correct sequence. That is done by the tRNA only. It is to pick up the amino acid from the, what you can say, for the polymerization, for the addition of what amino acid one by one. And it is picking up this amino acid from the cytoplasm of the cell only. And see the design, tRNA is what the normal RNA only, RNA is single stranded, but it looked like what a three-dimensional double stranded. See, it looked like what the double stranded. Why it is getting such a double stranded structure? It go for coiling. This is what the single stranded only. This is what the 5' end. Here you can see the 5' end. And this is what the 3' end of this RNA. It is single stranded, but it's go for coiling in a specific way to make it to perform its function. And in this coiling, what happened? And this complementary nitrogenous bases come close to each other. You may be not. It is not DNA, RNA only. Same like a DNA. Whenever these complementary nitrogenous bases comes together, it is having the tendency to make the hydrogen bond. So here we can find the bond between G and C, and it is what the hydrogen bond only. How many hydrogen bond between C and G? 3 hydrogen bond. And here we can find the bond between G and C, and A and U. How many bond? Only 2. And here UA, 2 bond, UA, 2 bond. In case of DNA, instead of U, T will be there. TA, 2 bond. Similarly, you also bind with the A with the 2 hydrogen bond. Here you can see what the bond formation between GC, and here we can see that hydrogen bond, and here also go for the hydrogen bond. Here it go for hydrogen bond because these are complementary, and here also it make a lot of bond between, because whenever that complementary nitrogenous base come close to each other, naturally it go for hydrogen bond, either 2 or 3. Depend on the nitrogenous bases and produce what the nitrogenous base pairs. And this structure, we can find so many stumps and loops. See, this is what the stump. And here we can see a round structure. This is what the stump. Here you can see the loop. Here there is a stump. Here there is a loop. And here you can see a loop. So here this stump is called as what the acceptor stump only. This is called as what the acceptor stump. Then we can find another stump here. It is called as what? The anticodon stump. It is called as what? The anticodon stump. You may be remembering the anticodon present in this loop only, right? This loop only. And this stump is what? The anticodon stump, and it is what? The acceptor stump. And here we can find three loops. Three loop. And this is called as what? The D loop. It is what? The D loop. Here we can see the D loop. This is what? The D loop. And opposite that we can find the T loop. Here can find the T loop here, and another loop present here, and it is called as what? The anticodon loop. Why it is called as anticodon loop? Because in this loop only anticodon is placed, and based on the nitrogenous base sequence in the anticodon sequence only decide what type of amino acid will bind here. Actually, amino acid is binding here in this 3' end. So always in the 3' end of all the tRNA end with the ACC, whereas in 5' end, the first one always should be guanine. In 5' end with guanine, and 3' end, ACC, and adenine only attached to the amino acid, and we call it as what? The amino acid binding site. Amino acid binding site. It is what? Amino acid binding site. And in T loop, here this is what? The T loop. And this is what? The T loop. Here you can see the T loop here. And this T loop only attached to the ribosome. There are two binding sites in a tRNA. So to understand the process of translation, you should know the detailed structure of the tRNA. Then only we will be able to understand how this amino acid attached to the tRNA, and how this tRNA attached to what? The ribosomes. Because at the time of what? This translation, this mRNA, I mean the tRNA, carry this amino acid to the site of protein synthesis and attach to the ribosome only, particularly in the receiving site. It attached with what? This loop, and we call it as what? The ribosome binding site. I mean, this is what? The ribosome binding site in the T loop only. So what is the function of the tRNA? How it's helping for protein synthesis in the cell cytoplasm? It is only to carry the 20 different type of amino acids. So to carry 20 different type of amino acid, different tRNAs are there. So we will see after discussing in detail about that genetic code, we will come to know about it. So total 61 different type of tRNAs are there in a cell cytoplasm, and by using this 61 different type of tRNA, it is possible to carry 20 different amino acids to the cytoplasm, to the site of protein synthesis. And this difference of all this tRNA is only the sequence of nitrogenous base present in the anticodon, and based on the anticodon sequence in the Vatican loop, anticodon loop, only determine what type of amino acid should be carried out by this tRNA, right? And come to the tRNA, very small RNA only. It consists of only less than 100 nucleotides. There are 61 different type of tRNA present in a cell cytoplasm, and each tRNA pick up a particular amino acid. To pick up a particular 20 different amino acids are there. To pick up that particular amino acid, there is a particular tRNA, and it is single-stranded, having 3' 5' direction, but it go for coiling and get a three-dimensional leaf shape, clover leaf shape, and 5' end is having guanine, and 3' end is having what? The ACC. Here you can see 3' end is always having ACC, and 5' end is having always this guanine only. And it coils to form three-dimensional clover leaf shape, 5' having guanine, and 3' end is having CCA. And 3' end is having amino acid binding site, and this end only bind the amino acid and carry to the site of protein synthesis. and it is also having an anticodon loop. There we can see the anticodon, 3 anticodon present in the loop, and that only decide what type of amino acid bind to this, what the 3' end of the tRNA, and anticodon sequence is complementary to the triplet codons. Maybe heard about the triplet codon, it is present in the mRNA. And every triplet codon is having its own meaning. And according to the nature or the sequence of triplet codon only, amino acid will be placed one after another by the tRNA, and codon and anticodon sequence are complementary. That triplet codon present in the mRNA, whereas anticodon present in the tRNA only. Triplet codon only giving message what type of amino acid should be carried. And based on that, according to the anticodon sequence present in the tRNA, it will go and pick up that particular amino acid and place in the that site of protein synthesis. So this sequence of amino acid in the protein is actually determined by the mRNA only, but based on this message given by the mRNA, tRNA only pick up that particular amino acid to the site of protein synthesis, right? So this is what the another machinery called as what the mRNA. Now we are discussing about the machineries only. Tell me, what are the machineries present in the cytoplasm for translation? rRNA is there, then tRNA is there, mRNA is there, and ribosome also there, then amino acids also there. tRNA, rRNA, mRNA, and the amino acid. These are the 4 machinery present in the cell cytoplasm, helping for the translation process. We have completed 2 machinery, rRNA and tRNA. Then come to the next RNA called as what? The mRNA. Why it is called as mRNA? mRNA means what? The messenger RNA only. This RNA is giving the correct message, and it is having what? The cap, and also having tail at the end. We already discussed about the processing of this RNA. In between the head, I mean the tail and the cap, we can find so many sequences of nitrogenous bases. And the continuous 3 nitrogenous bases present in this area only what? A codon, and we call it as what? The triplet codon. Why it is called as what? The triplet codon. In this codon, there are 3 nitrogenous bases, and it is code for a particular amino acids. And every triplet codon is having its own meaning. See here, this is what the Vatican say from the DNA. This mRNA is transcribed and having what? The tail and head region, and always the elongation of protein also take place from 5' to 3'. Remember, always the transcription is from the 5' to 3'. Then this translation also from 5' to 3', and even the DNA replication also always from the 5' to 3' direction only. In a biological system, this polarity is very, very important, always from 5' to 3' only. And see, these are the triplet codon given in the mRNA. So not like this diagram, there is no gap, there is no punctuation, there is no comma, there is no what you can say, such a type of things to distinguish one triplet codon from another triplet codon. So this the 3 continuous codon, I mean nitrogenous base only act as what? A codon. 3 nitrogenous bases are there in a codon. That is why we call it as what? The triplet codon. For example, in mRNA, GUA. G means what? Guanine, uracil, and adenine. And this acting as what? One codon. It's called as what? The triplet codon present in the mRNA. And this meaning of this codon is what? Amino acid valine. Right. So GUA, it is for valine amino acids only. The meaning of UGC present in the mRNA is what? Cysteine only. Then another lysine amino acids coded with what? This triplet codon AAG. and another serine amino acid is coded with the AGC, and CUU, it is for coding the leucine amino acids only. So every triplet codon is having its own meaning, and that only determine what type of amino acid will be placed one after another, right? And here then valine will be placed, and this triplet codon make the tRNA to bring the cysteine here, then lysine, then serine, leucine, and all these amino acids connected each other with a bond. You can see the bond here with given with the orange color. What type of bond connect one amino acid to another? Peptide bonds only. That is what the role of duty of what the RNA present in between the large subunits. So after discussing about the machinery required for this protein synthesis translation, we will go the process of translation. But you should know the role of every machinery present in the cell cytoplasm helping for translation. So 3 RNAs that we discussed, rRNA, mRNA, and tRNA. Then we will move to another machinery, and before that, see the triplet codon. I told you here, triplet codons are there in mRNA, and that only determine what the amino acids in a protein. And these are the triplet codons. Triplet codon, only 20 amino acids, but you can find 64 triplet codons. In mRNA, we can find 64 triplet codon, and this 64 triplet codon, out of the 64, and see these three triplet codons, UAA, UAG, and UGA, and these three triplet codons are not coding for any amino acid, and that only we call it as what the nonsense codon. Nonsense, no sense. All other codons are having its own sense. It is for what you can say, a particular amino acid, but these codons are not having any sense, no meaning. That is why we call it as what the nonsense codon, or we call it as what the stop codon. Why it is called as stop codon? Whenever such a triplet codon comes in the mRNA, naturally the process of the protein synthesis come to an end. That is why we call it as what the stop codon. Translation will be stopped whenever this triplet codon like UAA or UAG and UGA come into the triplet codon. So these three triplet codon we have to by heart as it is, because many chances are there to ask a question in the board examination with this concept. So this triplet codon helping for terminating the translation should be by hearted. It is UAA, no amino acid, then UAG, no amino acid, then UGA, no amino acid. And here you can see another triplet codon given in a red color. What is AUG? And it is called for amino acid called as what the methionine, and this is what the only triplet codon for the methionine. No other triplet codon for methionine, only one triplet codon, that is AUG only. It is having dual function. Dual function means to initiate the process of translation. In mRNA, first this triplet codon for placing the amino acid is what the methionine only, or AUG only. You can find so many triplet codon before AUG, and these are non-translated region. Such a triplet codon never translate. Always the process of translation start with the triplet codon AUG. So we call it as what the initial codon only. And not only to start, it is also for coding an amino acid called methionine. That's why I told you it's for the dual function, to initiate as well as for methionine amino acid. Then similarly, many triplet codons will be there even after the stop codon. Even after stop codon, many triplet codon will be there in the mRNA. and that region also will not be translated, and we call it as what? The untranslated region of mRNA only. So always translation between what? AUG and the stop codon UAA, UAG or UGA. So this is what the triplet codon, and we have only what four what you can say nitrogenous base in RNA, that is uracil, cytosine, adenine, and guanine. So with this, what three type of nitrogenous base only they are making what? The triplet codon. So three letters are taken. This is what the first letter UCAG, and it is what the second letter UCAG, and this is what the third letter, that is UCAG. So with this only we prepared what this table showing all the 24 different type of what 24 different type of triplet codon, 4 into 4 into 4, 4, 4, 4. So 64 triplet codons. Out of the 64, there is no meaning for these three, and we call it as what? The nonsense or stop codon, and methionine amino acid is coded with a triplet codon called as what? AUG. It is also for initiating the process of protein synthesis or translation. Then come to the next machinery, that is called as what? Amino acids. Another machinery is amino acids. See, in amino acids, we can find there are two functional groups. There are two functional. By using this functional group, only one amino acid connected to another amino acid with a peptide bond. And the protein is nothing but a long chain of amino acid. Thousands of amino acid will be placed one after another, and all these amino acids connect each other also with the peptide bond. Always connection between these amino acids. Functional group only, and there are two functional group. One is what? The amino group, and another one is carboxyl group. Amino group, NH2 group. Here we can see a functional group here, NH2 group. That is what? The amino group of the amino acid. And another one is what? The COH group. Here we can see COOH group. It is what? Another what you can say functional group, and we call it as carboxyl group only. This is what? The carboxyl group, and this is what? The functional group. By using this two functional group, amino group and carboxyl group only, amino acid bind each other with this peptide bond. And here we can see R, and the chemical nature of this varies from amino acid to amino acid. As I told you, 20 different amino acids are there in the amino acid pool in cytoplasm. Every amino acids are having these two functional group, that amino group and carboxyl group. And structure is what? The more or less same, difference only the chemical nature of this R attached here. Right? See, let us see how this amino acid connect each other with this peptide bond. This is what? One amino acid having the amino group, NH2 group, and here we can see COOH group, carboxyl group is there, and it is what? Another amino acid having what? The NH2 group, then carboxyl group here, COOH group here, and NH2 group of one amino acid connected to the COH group of another amino acid, and leave one molecule of water also. See here, OH group and H, these two, join together to form a molecule of water that drop from this bond and making a direct connection between this carbon and nitrogen. This will be taken out, direct bond between the carbon and nitrogen, and it is called as what? The peptide bond. Carbon and nitrogen, this bond is called as what? The peptide bond. This is what the way through which one amino acid connected to the another amino acids during the process of what? The protein synthesis in cell cytoplasm. That only we call it as what? The translation process. Right. So this is what amino acid, it's another amino acid, and many amino acid linked together. See, amino acid 1, amino acid 2, amino acid 3, like thousands of amino acids will be there in a protein. They are all linking here. by leaving one molecule of water in every bond. And finally, there is a link between this carbon and nitrogen. Carbon and nitrogen. There is a link between the carbon and nitrogen. And this bond only we call it as what? The peptide bond. Amino acids, this chemical nature may be varies. It's what and second type, or it may be the same type. The sequence of amino acids varies according to the nature of the protein only, or the use of the protein only. And in all cases, we can find only the amino acid link one after another and produce what? A long chain with the help of this peptide bond. And this long chain of these amino acids only we call it as what? The peptide chain, because it is connected with this peptide bond only, right? And this is what the amino acid. In a protein, you see the protein, a long chain of amino acids only. In every protein, one end with the free amino group, NH2 group is free here, and the opposite end of the protein is with the what? COOH group only. This is what the COOH group. So it is what? The carboxyl end of the protein. This end carboxyl group is free. So we call this end of the protein as what? The carboxyl end. And this end of the protein, thousands of will be there, not 3 or 4 or 5. Thousands will be there in a protein. In every protein, the first amino acid that is in the 5' end of the RNA only, first amino acid will be placed. And there NH2 group is free. And it is what? The amino end only. And the last, COOH functional group is free here, and it is called as what? The carboxyl end of the protein. That end is also very important for making what? The secondary structures, tertiary structure, quaternary structure, etc. Because the function of the protein is not only depend on the sequence of amino acids or the total number of amino acids present in the protein. Of course, that is very important. If sequence is changed, it cannot act as a functional protein in the cell. If number is changed, it cannot act as a protein in the cell. But at the same time, that that structure is also very important. For example, you maybe heard about the insulin hormone. It is working in our body to reduce the level of glucose only in its primary structure, not in the secondary or tertiary structure, right? Whereas in case of hemoglobin, it's a globular protein only. What is the function of the hemoglobin? Just to carry the oxygen from the lung to different parts of the body. But it performs its function only in the quaternary structure, not in primary, secondary, or tertiary structure. And actually, the primary structure will be converted into secondary structure through some kinds of coiling. Then secondary one go for again coiling to produce the tertiary structure. When this tertiary will go for coiling, naturally produce the quaternary structure. What will happen when we heat this protein? So naturally that bond will break. While coiling, it make a lot of bond, connect these amino acid each other with some kinds of bond. So bond will break. So what happened? It get. That is what we call it as what? The denaturation of protein by heating, right? And these are the 20 different type of amino acid. This we will represent it with a 3-letter code only. Here we have seen that 3-letter code. While discussing here, I told you that 3-letter code, GUA, 3-letter code only, right? GUC, there, it is called for amino acid VAL, this 3-letter code. So every amino acid represented with the 3-letter code only. So this is what the things given here, that 3-letter code. The alanine, ALA. Normally, the first 3 letters only taken to represent that particular amino acid. Alanine, ALA, arginine, ARG, aspartic acid, that is ASP. And sometime what happened, these 3 letters, for example, aspartic acid, ASP is taken, and asparginin, what the same letters only, ASP only, but it's already taken for the aspartic acid. That is why it's use another letter, ASN. And cysteine, 3 letters, glutamine, the 2 letters, Then glutamic acid, 3 letter GLU only, glutamine same letter only, but it is the last letter U instead, N is taken here. So glycine, GLY, like this is what the 3 letter code given for the 20 different kinds of amino acid. So 20 type of amino acids are there. These are the 20 different type of amino acid abundantly present in the cell cytoplasm. So we call this cytoplasm as what the amino acid pool. From our digestive system only, all the amino acids are being taken to the cell cytoplasm by the blood vessel, and amino acids are the product of the digestion of protein. Whenever we take the protein rich food material that undergo the process of digestion and produce amino acid, that only ultimately taken to the cell cytoplasm by our blood capillary. Some amino acid can also be synthesized in our body. So lot of amino acids are there in the cell cytoplasm, and it is what the raw material to synthesize different type of protein. Actually, what is the use of this amino acid in the cell cytoplasm? And by using this amino acid, only 20 amino acid, but plenty in number, by using these amino acids only, we are producing thousands of enzymes in the cell. Because just any amino acid can be used, and the sequence of amino acid, if you change, the nature of the protein also will change. Just like, as I told you, making what thousands of words in English with a limited number of letters. Same way, amino acids are like an English letter, and the protein is like many words can be synthesized, I mean, produced with a limited number of letters. Similarly, thousands of proteins can be synthesized with these 20 different amino acid. Thing is that the number of amino acid we have to change, and sequence have to change. Of course, that structure of the protein also can determine the function of protein in the cell. And these are all what the 20 different type of amino acids and their chemical nature. No need to go in very detail about all these things. Then come to the mechanism of translation. So now we have discussed the machineries. Machineries present in the cell cytoplasm helping for the process of translation. tRNA, remember the role and structure of tRNA, then rRNA, that ribosomal RNA only. See the importance of ribosomal RNA, then mRNA, messenger RNA, give the correct message for what type of proteins to be synthesized in the cell cytoplasm. And another one is what amino acid. And amino acids are the real raw material for preparing protein synthesis and for making proteins. And the 20 different type of amino acids are there. For every amino acid, we can find 2 functional group, amino group and carboxyl group, right? And see the process or mechanism of translation. Translation take place in the cell cytoplasm only because all the machineries are in the cell cytoplasm. And it is a process to prepare protein by using machineries. That only we call it as what the protein synthesis. It takes place in the cell cytoplasm. Machinery for protein synthesis are rRNA, tRNA, mRNA, and amino acids of 20 different kinds. And synthesized protein in the cell can act as enzyme, coenzyme, hormones, the carrier protein, contractile protein, etc. By using these biomolecule protein, cellular functions can be controlled. Cellular functions, all the chemical reaction, biochemical reaction or metabolic reaction happening in the cell is controlled by this protein only, because for every chemical reaction, enzymes are required. Enzymes are the biocatalyst. Without this catalyst, it is not possible to perform any reaction inside the cell. You may be heard about the nucleus. Nucleus is called as what? The controller of the cell. Controller. It controls everything. How this nucleus controls the entire functioning of the cell? Simply, nucleus only containing the DNA. And by using the DNA only, it transcribes the RNA. And by using RNA, it produces what? The particular protein in the form of enzyme. And this enzyme only the entire chemical reaction happening in the cell. That is why we call the nucleus as the controller. If, for example, A and B, these are the two reactants present in the cell. Every reactants are there in the cell. Normally, it never go for reaction. Whenever they want to go for reaction, then only it react. Why? Because A and B both are there in the cell cytoplasm, but they never go for reaction and produce C, right? But for this chemical reaction, enzyme is required. And enzymes only reducing the activation energy to make these two reactants to react, and it produce what? The final product C, right? So that is what the importance of what the protein synthesis. Then come to the steps. The process of translation. The first one, activation of amino acids. So plenty of amino acids are there in the cell cytoplasm. We call it as what? The amino acid pool. But to make this amino acid to prepare a protein, we have to activate the amino acid present in the cytoplasm. That is what the first step, activation of amino acid. Actually, protein synthesis is what? Energy required process. Energy is required to synthesize protein in the cell. And what is the need of energy for protein synthesis? Mainly for activating the amino acid. See, ATP molecule, it is very much required here, right? ATP molecule is very much required here. See. Right. Activation of amino acids, something went wrong. Yeah, see, it is what? Energy required process. For activating an amino acid, this amino acid present in the amino acid pool react with the ATP molecule with the help of the enzyme and produce what? Amino acyl. Amino acyl is amino acids only. Bind with AMP. ATP will be converted into AMP and release 2-phosphate molecule. AMP means what? The monophosphate only. And it is what? The triphosphate only ATP. So amino acid bind with the ATP molecule with the help of the enzyme and produce a complex amino acid or amino acyl AMP enzyme complex and 2-phosphate molecule base. Sorry, 1 minute. Huh?
We are going breakfast today to me. I will in biology lab.
Yeah. So, sorry. Then this is what the first process, activation of amino acids. This amino acid to activate first bind with ATP with the help of the enzyme and produce a complex. This complex consists of amino acid, AMP and enzymes. Enzymes are catalysts only that will be released later. Amino acid bind with the ATP, form amino acyl AMP enzyme complex, and it is monophosphate only here, AMP, and 2-phosphate molecule will be released. This is what the process of activation of amino acids. Here you can see this is what the amino acid, and it is what the adenosine triphosphate, 3-phosphate molecules are there, 1, 2, 3, adenosine triphosphate molecule. This is what? Adenosine, adenine, then pentose sugar, ribose sugar only, 3-phosphate molecule, adenosine triphosphate molecule that connected to the amino acid and form what? Amino acyl adenylate, and only what? 1 phosphate molecule here, and here 2 inorganic phosphate released. This is what the real process of what? Activation of amino acid. This amino acid present in the amino acid pool should be activated by using ATP molecule. Then only that amino acid can be used for the protein synthesis, right? Because all the amino acid present in the amino acid pool is not active, very inactive. Only after activation it can be used for protein synthesis. And second one is what binding of amino acid with the tRNA. See, after the activation of this amino acid, it bind with the tRNA. tRNA means what? The transfer RNA. So amino acyl AMP enzyme complex chemically bind with the tRNA. This is what the reaction between the tRNA. Remember that structure of the tRNA is having what? Amino acid binding site is at this 3' end. Right, that amino acid, this amino acid, activated amino acid only bind with the T. Activated amino acid is nothing but amino acyl AMP enzyme complex. That is what the activated amino acid that bind with the tRNA to form amino acid tRNA. AMP will be released and enzyme also will be released. It is ATP and enzyme only to activate and only to produce what this amino acyl AMP enzyme complex. But after meeting it with the tRNA, tRNA means what? There is a particular tRNA for binding a specific amino acids. Then after meeting with the tRNA chemically react with the amino acid and in the 3' end form amino acyl tRNA complex, and AMP will be liberated and enzyme also taken out, and amino acyl tRNA is called as what? Amino acyl tRNA. This tRNA, amino acyl tRNA. Amino acyl means amino acid only. It is what? The activated amino acid bind with the tRNA is called as what? The amino acyl tRNA. This is what? Amino acyl tRNA. AAtRNA, amino acyl. Amino acyl means what? The amino acid only, it is connected to the tRNA, but it is possible only after the activation of amino acids and form amino acyl tRNA. Right. Here you can find this. What is this? tRNA. Transfer RNA. Why it is called as what? The transfer RNA, because that only transport the amino acid to the site of protein synthesis. And here you can find this 3' end, 3' end. Here in 3' end is called as what? Amino acid binding site. And this tRNA bind with the amino acid. This is what? The amino acid. This is what? The amino group, and this is what? The COH carboxyl group. And it bind with this carboxyl group only. Right, the carboxyl group, this group of the amino acid only bind to what? The 3' end of the tRNA, exactly like what? The peptide bond only. Here in 3' end, adenine is there having what? NH2 group, and this amino group of this 3' end, what you can say, make a bond with the carboxyl group there. Just like what? The peptide bond we have seen. So this is the way through which these tRNA react with amino acid to form what? Amino acyl tRNA. And this is what? The anticodon. Anticodon means what? 3 codons will be there, and 3 nitrogenous base will be there. And this nitrogenous base is what? The complementary to the triplet codon given in the mRNA. And that only decide what type of amino acid should be bind there in the 3' end. So 20 different amino acids are there, but these 20 different amino acids can be coded with the 61 different type of triplet codon. Triplet codon 61, and 3 are not coding for any amino acid. So these 61 triplet codons are having what? The 61 anticodon. So 61 different type of tRNAs are also there. And difference is that what? The difference in the anticodon sequence, only 61 different type of tRNAs are there. For every tRNA, there is a particular amino acid that only decided by the triplet codon present in this what? The loop. We call it as what? The anticodon loop of what? The tRNA only. Right? Then come to the third one, initiation of polypeptide chain. First one is what? The activation of the amino acid present in the pool. Second one is what? The binding of the activated amino acid to the tRNA. Then third one is what? Initiation of polypeptide chain. Initiation, starting of this protein synthesis. Here, you can find this is what the mRNA. This is what the mRNA sequence. In mRNA, we can find so many nitrogenous bases only. So many nitrogenous bases. As I told you, there is no comma, there is no full stop, there is no gap in between. But I have given here with the three colors. That is to identify what the triplet codon present in the nitrogenous base, present in the mRNA. For that only I have given different colors, given the triplet codon with the black color as well as in the red color, right? So always the process of translation start with the AUG only. While discussing about triplet codon, I told you AUG for dual function. What are the functions of AUG? One is to code for methionine amino acid, and another one is to initiate the process of translation. And many triplet codon will be there before this AUG. Here you can see start. Before AUG, many triplet codons will be there. In the cap region, that is what the 5' end only. From 5' end only, what the what you can say, that process of protein synthesis starts and elongate from 5' to 3' direction. This is what the direction. And here we can see a cap, and here we can find a tail. Right. But always remember, this protein synthesis starts from the AUG. That is what the starting triplet codon for the methionine amino acids. Many triplet codon will be there before that. And this region are called as what? The untranslated region at the 5' end. At the 5' end. Many triplet codons will be there before AUG, and these are called as what? Untranslated, because this region never get translated. Translation always start from the AUG only, and not from any other triplet codon. So triplet codon present in the 5' end of this mRNA before AUG is considered as what? The untranslated region in 5' end only. And similarly, here we can see a stop codon. That is UAG, stop codon, UAG, codon or stop codon only. Even after the stop codon, you can find so many nitrogenous bases. And these nitrogenous bases are not working as a triplet codon, never code for any other amino acid, because with this AUG, what the real process of protein synthesis will stop. And this nitrogenous base sequence present in the 3' end of this mRNA is called as what? The 3' UTR. What is UTR? Untranslated region. This region also never get translated. mRNA, this is what the mRNA. Now we are discussing about the initiation, how the process of translation initiate. Starting always with the AUG. Then after starting, according to the triplet codon given the mRNA, amino acid will be placed one after another. For example, AUG, the first one starting, methionine will be placed. And next one, UUU, it is for what you can say, phenylalanine. So phenylalanine will be placed. And another one is CUU, leucine will be placed. Then AUG, again, methionine will be placed. So it can be repeated many times according to the nature of the protein. You don't think that AUG is only for starting. No. Of course, every protein start with methionine amino acids only, but in between also it will come depend on the nature of the proteins. Right. So according to the triplet codon given in the mRNA, tRNA will come and place all the amino acid one after another. So triplet codon and anticodon, remember. For example, to carry that methionine amino acid to the site of protein synthesis, the triplet codon is AUG. What is the anticodon present in the tRNA? What is the anticodon present in the tRNA? If it is A, first one should be U. If it is U here, second one should be A, and G, it is C. So UAC, that is what the anticodon present in the tRNA. that tRNA only able to carry amino acid to the site of protein synthesis. Remember that loop at the bottom of tRNA called as what? The anticodon loop. In that loop, there are 3 codon, and we call it as what? The anticodon only. And anticodon is complementary to the triplet codon given in the mRNA. See, in case of methionine, only one triplet codon, that is AUG only. So always which tRNA carry the amino acid methionine to start or to place methionine in between? No doubt, these are the tRNA having an anticodon in the anticodon loop, and this anticodon C what? UAC. That only will, that is what the complementary to the triplet codon present in the mRNA. And based on that triplet codon in the mRNA, that particular tRNA with what you can say complementary anticodon will go and pick up the amino acid only after activating the amino acid by using what? The ATP molecule from the amino acid pool, and it bringing to what? The site of protein synthesis one after another, and it will be placed. This amino acid will be placed one after another by the tRNA by seeing what? The sequence, the triplet codon present in the mRNA. Always it start with the AUG, remember, and end with the stop codon or nonsense codon like UAG. So number of number of amino acid present in the protein depend on the number of what? The triplet codon sequence present between the untranslated region of 3' and untranslated region of the 5'. But there is no amino acid for the stop codon, that is also there. So should not calculate the stop codon for counting the total number of amino acid present in the protein. AUG you have to count, because it is not only for initiation, it is also for methionine amino acid. But that the last codon, triplet codon, stop codon, never code for any amino acids. So come to the initiation of polypeptide chain. Starting of polypeptide chain, mRNA only, not RNR. mRNA occurs in between large and small subunit of ribosome. In between, mRNA is what? Very elongated. In that only that message is given regarding the type of amino acids will be placed, and it occurs in the between the large and subunit of ribosome. Ribosome is having that rRNA. mRNA consists of nitrogenous base sequence. This sequence only decides what type of amino acid will be placed one after another, and 3 nearby base code for a particular amino acid called as what? The triplet codons. 3 nitrogenous bases in the mRNA is acting as what? A code for a particular amino acid. We call it as what? The triplet codon. In mRNA, sequence are in a regular fashion. Regular fashion it is given, no comma, no full stop, no gap, nothing. Is what? Nitrogenous base. It is what? Given in a continuous fashion. The process of initiation of translation with the triplet codon AUG, and this AUG triplet codon is called as what? The initial codon, and AUG triplet codon is also called for methionine amino acid. And the process after initiation of what? The translation, tRNA will place all the amino acid one by one according to the triplet codon present in the translation region. And whenever any one of this codon comes in the mRNA, triplet codon comes in the mRNA, UAA, UGA, and UAG, by heart the triplet codon for the termination of translation, because there is no amino acid code for this triplet codons. And that is why we call it as what? All other triplet codons are having meaning. It is not having any meaning, not called for any amino acid. So we call it as what? The stop codon, UAA, UGA, and UAG. So I told you here, it is given in a continuous fashion. There is no full stop, gap, or anything in between. I have given 3 different colors only to distinguish it, to understand the concepts. But there is no such a gap or color difference or comma or full stop, nothing in between. It is arranged, all the nitrogenous base arranged in mRNA in a continuous fashion only. But all are having meaning. See, this AUG is having meaning, and UAC is also having meaning. UAC is also having meaning, but it is not translated. Why? Because it is located just before the starting codon only. So first amino acid in all protein are what? The methionine only, because it is what? The starting codon for the translation process, right? See, this is what the what you can say, mRNA, and it is from 5' direction only. So translation extent from 5' direction, and it is moving in this direction only, from 5' direction to what? The 3' direction only. And there are this mRNA located in between what? These large subunit and small subunit of ribosome. In this junction, we can find the rRNA, and this rRNA only making the bond between these amino acids with the peptide bond, and after keeping what? The amino acid according to the triplet codon given in the mRNA, normally ribosome slowly move in this direction from 5' to 3' direction. There is a gap between large and small subunit, and in between that mRNA will be there. Through this mRNA, it is moving from the 5' direction to 3' direction. So that only helping for the elongation of amino acid. This tRNA will place what? Amino acid one after another. So naturally they get elongated. And whenever it come to what? The nonsense codon or the stop codon, with that the process of elongation will be terminated. So this is what the way. So this is what the amino acid, and first initial initial triplet codon is what? The AUG, and it is called for amino acid. So the first one, first tRNA having an anticodon UAC, taken that amino acid methionine and place it here, and second one UAU, a triplet codon, and this is what the tRNA having UAA anticodon carry another amino acid, will place it here, and there is a RNA here, that there is RNA here in the ribosome called as what? The rRNA, that only making the bond, and one by one according to the triplet codon given, because it is moving. Whenever it is moving, this triplet codon will enter into that ribosome. So naturally, one by one amino acid will be placed according to the triplet codon given present in the mRNA, and it make the peptide bond, and it will elongate. So this is what the way through which it is what? Increase its length. That only we call it as what? The elongation. So it go for elongation in this direction. So that only we are going to discuss in very detail here. That initiation we have discussed. Then let us see another one. This is what the initiation. Then we will go to what? Another one. See, the tRNA having UAC anticodon. Take the methionine, because the methionine amino acid is coded with the triple codon, that is AUG only, and this is what the anticodon UAC. And it take the methionine amino acid from amino acid pool after activation to the site of translation by using what? Amino acid binding site of tRNA. Remember, there is a site in 3' of this tRNA, where this amino acid will bind after the activation. Then activated amino acid tRNA attached to the P site of the ribosome. That is what the peptidyl site or the donor site only. Here you can find the donor site here. Already we discussed here. Right. And this is what the first amino acid plays only in the P site, not in the A site. A site is what the acceptor site. But to initiate, methionine only is starting this amino acid that will place, the first one will place first in the P site only, not in the A site. From P site, it will be taken to E site. From E site, tRNA will come out. Then that time what happened, second amino acid onwards, tRNA will be placed in the A site only. The first tRNA carry the methionine attached to the P site only, and second one onwards, all the tRNA will attach to the A site only. So whenever ribosome move from P site, this is the first one move to E site. So from now the P site is free, A site is free. So we come to the A site. Then whenever it move, it come to the P site, A site is free, next one will be attached. So all the time, whenever the ribosome move, the A site is getting vacant. Whenever it is vacant, naturally, according to the triplet codon given in the mRNA, tRNA will place what? New amino acid. And whenever it move from the A site, it move to P site. From P site, tRNA will move to the E site. And after that time, it will connect. In the P site itself, what happened? All the amino acids connect with that peptide bond and it elongate. And from the E site, it is called as what? The exit site. And from the E site only, the tRNA will taken back to the cytoplasm. Then again, this tRNA will attach to this amino acid after the activation. Right. So this is what the things mentioned here. Another tRNA carries second amino acid according to the first one, the activated amino acid tRNA, that is attached to the P site of the ribosome only, or it's called as what? The donor site or peptidyl site. P site, peptidyl site. And another tRNA carry the second amino acid according to the information given in the mRNA triplet codon, and this tRNA attached to the A site. First one is attached to what? The P site only, and another one, this is what? The methionine attached here, and second one attached to the A site. Now A site is attached with the tRNA and P site is also attached with the tRNA. Whenever ribosome move, naturally from the P site, tRNA is taken to the E site, and from the A side, it is taken to the P side. Whenever this tRNA move, naturally along with that amino acid is attached to what? This tRNA only, right? So this is what the thing mentioned here. Another tRNA carries second amino acid according to the information given in the mRNA as a triplet codon, and this tRNA attached to A site of the ribosome amino acid site. That is what the what you can say acceptor site only. So second one attached to the acceptor site and first one attached to the P site. Now initially we can find there are two tRNA attached to the ribosome. One is first one attached to the P site, that is always what? The methionine only. Methionine carrying tRNA only, and second one attached to the A site. Then whenever the ribosome move forward, these two tRNA taken to the next site. From A site, it is taken to the P, and from the P, it is taken to what? The E site only. Whenever it is taken from the P to E, that tRNA detached from the amino acid, and after detaching, it's connected with what? The second amino acid. So like that, it go for elongation. And this is what? The methionine amino acids. And here, to initiate the process of what? The protein synthesis, triple codon AUG only helping. It is what? The initial codon. And this tRNA having UAC anticodon, because that is complementary. That should be complementary. That triplet codon and anticodon should be complementary. So AUG, its meaning is what the methionine amino acid only, and this tRNA having UAC triplet code, anticodon, will attach this methionine amino acid in its 3' and it is only taken to what? It is only taken to the site of protein synthesis to initiate, to start the problem, start this what the translation program, right? And come to the elongation. As I told you, always protein elongate its length from 5' direction to 3' direction. See, RNA, mRNA is having its own polarity, having 5' end and 3' end. Always the first amino acid will place in the 5' end of this mRNA only, and it's moved towards what? The 3' end. So elongation of this protein always from the 5' 3' direction of the mRNA. According to the triplet codon given in the mRNA, amino acid will be placed one after another because all the triplet codons in the mRNA is having its own meaning. Meaningless but you can say triplet codons are taken during the process of what? Processing. That is what the introns are taken. Exons are having the meaning. So will be placed one after another. After placing the initial what you can say amino acid called as what? The methionine. And after bringing the third amino acid by another tRNA, ribosome move little forward. So first of all, ribosome is there. There are three binding site there. A site is there, P is there, and E site is there. And first two amino acids, the first one is what? The methionine only placed at the center. That is what? The P site only. And second tRNA carry another amino acid and place in the A site. Now E site is free. Now what happened after placing these two tRNA on this A site as well as the P site, ribosome move little forward. So what happened? From P side, tRNA move to E side, and from A side, pRNA move to what? This side. Sorry. So this is what the process happening during the process of elongation. The first amino acid is methionine carried out by a tRNA, and this will be placed in the middle P site only, donor site only, and second amino acid carried out by another tRNA placed in the A site only, the first one. Then after saturation of these two sites, the ribosome move little forward. So what happened? From the P side, tRNA is taken to the E side. From A side, the tRNA is taken to the P side. So while taking the tRNA from the P side to E side, it detach it from the amino acid. Now the amino acid one functional group is free, and this functional group is attached with what? The functional group of the second amino acids. Then like that it will move little forwards. Now the A site is free now. So to the A site, another tRNA will bring amino acid, and similarly what happened? It move little forward. So or normally whenever it move, you know that A site is free, and in that free A site, new amino acid will be placed by the tRNA. But all the time that P site is having what? The P site and E site after initiation is having what? The tRNA. Whenever the ribosome move forward, actually A site is getting free, and this free A site always receive. That is why we call it as what? The acceptor site. All the time it accept what? New amino acid, the tRNA only. So initiation only P site is used for attaching this tRNA with the methionine amino acid. Afterwards, A site only accept and receive what? This tRNA with these amino acids. So that the first amino acid will come out from the ribosome, and the first tRNA connected to the amino acid will fall from P site to the cytoplasm. See, after bringing third amino acid by the another tRNA, ribosome moved forward little, so that the first amino acid will be come out from the ribosome. That is what the elongation process. And the first tRNA connected to the amino acid with will fall from the P site. That means from the E site, the tRNA is not connected to amino acid, that fall to the cytoplasm, and the second tRNA move from the A site to P site along with the ribosome attached. So always E site, it is called exit site. From exit site only this tRNA is taken out, and that time whenever the tRNA comes to the E site, there will be no ribosome, because this there will be no amino acid, because this amino acid detached from the tRNA and connected to what the another amino acids present in the P sites. So like that, added taken tRNA one after another to the A site according to the triple codon given in the mRNA. Whenever it take the tRNA, it carry the amino acids also, respective amino acids also. This also will be link each other with the help of the peptide bond, and slowly it go for elongation. Here, this is what the A site, P site, and E site present in the ribosome. So for starting this process of what the protein synthesis or translation, you see AUG is what the initial codon. So the first tRNA having UAG, UAC anticodon carry a methionine amino acid, and that will place first only in the P site to initiate. Then here you can see another triple codon UGG. So that tRNA having what ACC anticodon carry what another amino acid, that tryptophan, and it is taken to what the A site. Now there are two what you can say tRNA attached to the ribosome, P site and A site. P site only for initiation, and second amino acid is taken to the A site only according to the triplet codon given in the mRNA. Then after keeping these two amino acid in P site and A site, what happened? It moved little forward. So another triplet codon will enter into the ribosome. So while moving this ribosome towards what the 3' end, this is what the 5' end, and it is 3' end. I told you elongation is always from 5' end to 3' end only. And whenever it moves from the 5' to 3' end, what happened? The tRNA present in the P side moved to E side. Whenever it moved from the P side to E side, what happened? This methionine will detach from this the tRNA, and this functional group of the methionine connected to what another amino acid placed in the A site only. So what happened? This tRNA reached to the E site. Now what A site is free, because from the P site tRNA is moving to E site. From A site this tRNA is moving to P site. Now A site is free. So what happened? Next amino acid will be taken according to the triplet codon given in the mRNA, because it is inside the ribosome only mRNA. So based on the triplet codon, next triplet codon, the another amino acid will be taken by this tRNA and keep it in the A site. So what happened to the next movement? Now all these three sites are having that this tRNA. A is having, P is having, and E is having. So what will happen? E, you know that the tRNA present E is not connected to methionine, any amino acid, but the tRNA in A and P sites are connected to the amino acids. Whenever it move little forward, another one will be placed. So what happened? There will be a movement of what the tRNA from A site to P site, then P site to E site, and whenever it moves, no P site tRNA is alone, not connected to ribosome. So what happened? From the E site, it will go directly to the cytoplasm. That is why we call it as what the exit site. Exit. That tRNA comes from this ribosome and goes to this cytoplasm, and this tRNA again can go for binding another amino acid for same proteins that is there. So this is what the process of elongation. So whenever it is placed one after another, this amino acid by these different kinds of tRNA, naturally there will be what? Elongation. So whenever it plays, this will be pushed little. Another one plays, it pushed little. So finally what happened? This protein will be coming out from the ribosome in the form of a long chain of amino acid connect each other with this peptide bond only. This is what the process of elongation of this protein during this protein synthesis in the ribosome. So from the ribosome only protein get elongated and come to the cytoplasm. That is why we call this ribosome as what? The protein factory. Because it is only producing this, it is only producing the protein with the help of what? The rRNA, ribosomal RNA present in the junction of the large and the small. This is what the next one will bring it here. So from the E side, the tRNA is taken out. So naturally that methionine amino acid connected to what? This what? The first tRNA linked with the second one. So another one will be taken to the A side. Now A side is free. So it is connected here. So what will happen? Next, again, it will move little forward. The ribosome move little forward. So during its movement, what happened? Another triplet codon will enter inside. So from A side, it will move to P side. Always from the P side, it will be taken to E side. So there will be an ejection, there will be an excess of this tRNA from the E side to the cytoplasm. So this movement only helping the protein to elongate its length, and finally one by one, these all amino acids will come in the form of chain, and it is called as what? The polypeptide chain, and formation is always from 5' to 3' direction only. And it is taken and it is moving outside. So this is what the things mentioned here. So this will continue by reading the triplet codon present in the mRNA. mRNA consists of what? The sequence of many number of triplet codon. They read all the triplet codon present in the mRNA. During elongation, new amino acid will be placed one after another in the 3' end of the mRNA and increase the length of polypeptide chain, and it is called as what? The protein. This polypeptide chain is called as what? The protein. Amino acid molecules of the constructed protein comes out from the ribosome one by one. Whenever we place, it will come out from the ribosomes. A new one is added, another one will come out. So because whenever new ribosome amino acids are added to that protein, the amino acids also come out from the ribosome one after another, and peptidyl transferase enzyme connect one amino acid to another amino acid with a peptide bond with the support of the rRNA. I told you about the role of the rRNA. But the rRNA is acting as what? An enzyme only. You know, beginning we have discussed about the RNA. It's not like a DNA. It is acting as what? The biological catalyst. That is why it is what you can say, not much stable. And peptidyl transferase enzyme, and that only connect what? One amino acid to another amino acid and produce the peptide bond with the support of the rRNA. Like that, it is taken one after another. And this ribosome is moving forward from 5' direction to 3' direction. Whenever new whenever new amino acid is added, and whenever it moves, naturally what happen? One by one, this amino acid will come out from the ribosome, and it will be elongated and connect this peptide bond, and it is what? The growth of the peptide joint. It is moving, and whenever tRNA is used here for constructing this protein, one by one this tRNA will come to the cytoplasm, and afterwards this tRNA can go and pick up another amino acid. Here it is having ACZ, so naturally this can go for picking another amino acids with what you can say triplet codon, that is called as what? The that is what? UGG triplet codon. Here UGG, first triplet codon, right? It is not methionine. It is taken some other amino acid, and this afterwards what? Due to the movement, this will be separated, and this can be activated again and can go for bringing another amino acid to the site of protein synthesis. So this is what the process of elongation. Then come to the termination. What is termination? The process, elongation come to an end. We already discussed there is a triplet codon for stopping the process of what the protein synthesis, and we call it as what the nonsense codon only. So in mRNA, there are some triplet codon which are not coding for any amino acids. Such a triplet codons are called as what the nonsense codon or terminator codon or stop codon. Call it as what the terminator codon or nonsense codon or stop codon. Why it is called as nonsense codon? Because it is not having any meaning. It is not meant for any kinds of amino acids, and it is a terminator codon because it stops the process of what the protein synthesis, translation. There are three types of such a triplet codon identified. That is what UAG, UAA, and UGA. This triplet codon we have to by heart, find out some techniques to by heart what the triplet codon to stop and also triplet codon to start. AUG, that is what the initial codon, and UAG, UAA, UGA, and C is not there anywhere in the terminated codon. Whenever it comes in the what you can say, whenever any one of the terminated codon comes in the mRNA, the process of the translation will come to an end, because there is no tRNA having that triplet anticodon. See, UAG, it's what the triplet codon given in the mRNA, but there is no what you can say tRNA having an anticodon AUC, AUZ. It is what the complementary anticodon for the tRNA, and such a type of tRNA is not at all there in the cell cytoplasm. And there is no tRNA having the anticodon AUU, and there is no there is no tRNA having that is what ACU, that is also not there. So no tRNA to bring any kinds of amino acids. That's why I told you these are all meaningless type of triplet codons only present in the mRNA as a nonsense codons only. So there is no tRNA to carry such any kinds of amino acid to the site of protein synthesis. So the process of elongation come to an end. After completing the protein synthesis, ribosome unit also separate, because the mRNA present in the ribosomes only between the large subunit and small subunit. So what happened after completing or terminating the process of translation? These two subunit, large and subunit, detach, and it will detach to the cytoplasm and drop into the cytoplasm. In cytoplasm, again, it go for rejoining, and it can also go for protein synthesis later, right? So this is what the protein synthesis ribosomes after the termination, and it this is what the elongation, and these are the what you can say protein synthesize and come to an end due to the nonsense triplet codon present in the mRNA. After completing the process of protein synthesis, the large subunit separated from the small subunit, and it will drop into the cell cytoplasm. Again, it get activated and joined together, and this can be act as what functional ribosome for another protein to synthesize. So this is what the process of bringing this methionine amino acid to the mRNA to initiate the program. See, initiation, these are all non-translated region only, because always protein synthesis start with the AUG. So to this side, this tRNA having anticodon UAC bring the methionine amino acid and place it here, and remaining triplet codon is also having their own meaning according to the triplet codon given in the mRNA. this tRNA, 61 different tRNAs are there. It can bring 20 different amino acids. So according to the triplet codon given in the mRNA, tRNA will bring the amino acid according to the triplet codon in the mRNA, place one after another and produce what? A long chain of amino acid, connect each other with the peptide bond. And that only we call it as what? The polypeptide chain, or we call it as what? The proteins only. So this is what start codon and produce what? The small protein. And this if for making a new long protein, not only required what? One the ribosome, because every ribosome is having its own limitation. After making some chain, it loses its what? The enzyme power. So naturally what happened? It will be there, another ribosome will attach and it's make the remaining part, and another one after making some chain, no, make another one will be attached and it make what? Another one. So it make this protein piece by piece only, later will be connected each other with the peptide bond to form a long chain. And sometime whenever we observe this cell under microscope, we can see a structure like this. One mRNA connected to many ribosomes. Many ribosomes will be there in one mRNA. Why? Because ribosome is having its own limitation. After making the chain or a particular length, naturally it reduce what? The power of its enzyme. So another ribosome will attach to the mRNA, do the same work, and bit by bit only they are making what? This protein here. Later it will be joined. At a time we can find many ribosome attached to the mRNA. It is just like what? Rough endoplasmic reticulum. Sometimes it looks like what? The rough endoplasmic reticulum only, but it is not rough endoplasmic reticulum. The thing present at the middle is nothing but the mRNA only, and this structure only we call it as what? The polysome or polyribosome. It is not rough endoplasmic reticulum. Sometimes whenever we observe it with what you can say, some kinds of highly magnifying microscope, we will able to see what? mRNA connected with the many ribosomes. So at the end, what happened? All the ribosome will detach and it will go to the cytoplasm only, right? And this only we call it as what? The polysome or polyribosome. Poly means number of ribosome attached to the mRNA. So as I told you that nature of the protein or the function of the protein is determined by total number of amino acids present in the protein, then sequential arrangement of amino acids present in the protein. If the number of amino acids in the protein is less, that will not function as what? That will not work as what? Functional protein. They cannot do any work in the cell. If sequence is changed, naturally that also affect what? The function of protein in the cell as an enzyme. So every enzyme required the correct number of amino acid in the protein, and sequence is also very, very important. And not only these two, another factor to determine the function of this protein in the cell is what? Its structure only. Structure can also determine. As I told you in the beginning, some protein work in the cell only in the primary structure. Some are only in the secondary structure. Some are in tertiary structure. Protein work in the cell as a functional protein only in the quaternary structure. See, insulin work as a hormone in its primary structure in our body. Two chains are there, maybe remembering A chain and B chain. The American company Lilly, they produce what? The A chain and B chain separately. That we will see later while discussing about what? The another chapter, that biotechnology, they made what? Insulin artificially. Two chains. Because C chain is also there. A, B, and C. C-shaped chain connect A chain to the B chain. It is what? The primary structure only. And secondary structure, alpha helix and beta plated sheet. Then just go for coiling, primary structure, just these are all what? Round one. Amino acids only go for coiling and getting what you can say, secondary structure, tertiary structure, and quaternary structure. Whenever a particular amino acid, some are having the positive charge, some are having negative charge, and hydrostatic, what you can say, force of attraction. So many things were here to connect one. Whenever it is going for coiling, there will be some kinds of connection between what the amino acid present in the protein due to what the different reason like charges. That only making them to get what you can say a three-dimensional structure whenever they go for coiling to get a structure like this. Primary, secondary, tertiary, quaternary structures. And that only determine the functions of the proteins. So as I told you, which determine the function of protein prepared in the cell as an enzyme or as a hormone, as a carrier protein or as contractile proteins. One is a total number of amino acids present in the protein. That should be very correct. Then sequential arrangement of amino acid in a protein. That is also very important, right? It is very important. It is just like my name is Minal. If that spelling, if it is MNIL, its meaning will be different. So that sequential arrangement of amino acid is in the protein is also determine what the function of protein in the cell. Then come to the structure. Some are in primary structure, some are in secondary structure, some are in tertiary structure, and some other protein work in the cell only in its quaternary structure. So structure is also very important. And such a type of protein can act as an enzyme or hormone in the cell to change or speed up the chemical reaction within the cell and depend on the temperature and pH. For every enzyme required optimum pressure, optimum temperature and optimum pH to work or to work as what the catalyst to make many that material to react inside the cell. So these are the three things determine the function of protein in the cell. Then we will move to another one after a break, and we will start our next section at 2:30, and there we will discuss more detail about this genetic code. So now you can move for your breakfast and come and join with me at 2:30. So we will have a afternoon genetic code only. So thank you so much. Now you can move for your breakfast. Thank you so much. Last day, sir. Last day. Good. But we are not getting the response now. That's the problem.